All right, today we're going to be looking at antibiotic resistance. And so what we're talking about here is antibiotics that break down, and I'll show you in a second an image, they basically disrupt how bacteria functions. So they're antibiotic, which means they are anti-living things, which bacteria is that living thing. And so what's happening here is over time, we've had quite a few antibiotics that have been created to sort of stop these bacterial infections and diseases, but they're losing their ability to do that job. And so the way that they're doing that is they're going through a process of adaptation and evolution where they're changing some of their gene sequences so that they can combat even our harshest antibiotics. And so this lab in particular is going to be us using these antibiotics on a strain of E. coli. E. coli is probably one of the most famous bacteria there is. Every few years there seems to be an E. coli outbreak. The E. coli that's used in this lab actually is from a human, or it's not dangerous to human beings in that it is a type of bacteria that we have in our stomachs already. But you may have heard of restaurants being shut down for an E. coli outbreak where that E. coli from a cow or some other animal that we consume brings bacteria into our bodies and makes us sick. And so I'm going to show you the sciencey bit right now. So if we go ahead and take, yep. So what we see here is there are multiple ways that antibiotics can stop a bacteria from functioning. So you can see if we start at the very top there, you can see it can stop transcription. Transcription is the process by which DNA is written onto an RNA molecule. And so it can stop that from happening. It can also stop translation. Translation would be when the protein is being produced from that transcribed piece of DNA. It could stop the enzyme from doing enzyme action. Remember, enzymes either break down or put things together, so it can stop that process. It can break the membrane of the bacteria, or it can stop it from replicating, so it can't make more copies of that bacteria. And so these are all the ways that we could have that antibiotic work to get rid of a bacteria we do not want. So this is a chart here that you're going to see that is going to have what the plate's going to look like when we're all done. So once we have put our discs, these are, they almost look like paper punches that have antibiotic on them. And we're going to put them on an E. coli plate, and I'm going to show you how to build the plate and how to put it all together. And so you can see that there's a circle around each one. So 6 obviously has a bigger circle than 1. And so what that's showing me is that there is resistance. The bigger that circle is, the more resistance there is. So if there is no circle, like on number 4, that means that the antibiotic does nothing. That could even be our control. It could literally be a paper punch that's been put there and it's not causing the bacteria to be moved away at all. So those are spots where the bacteria does not live. And so you see a clear sort of window of where bacteria should grow but does not grow.